Yeah, thank you very much for the introduction. Thank you for the opportunity to present a little bit about the BovReg project. So you'll hear a little bit about the general overview of the project, and then I think we selected to give some results to give you a flavor of what we achieved. So it will be not very comprehensive, but it will pick out individual examples. BovReg, as you probably know, is a consortium comprising 20 partners across 13 different countries from Europe and also from Canada and from Australia. BovReg, the essential target of BovReg is to annotate the Bostaurus genome with a particular emphasis on traits like health, robustness and biological efficiency, which also drove selection of populations, samples and traits we monitored. Among the several objectives we had in BovReg, you'll hear about three particular in the next minutes and the next two talks. This is to annotate the functional active genomic regions in the bovine genome, to map molecular and whole genotype phenotypes in the bovine genome, and then also to develop prototype models for integrating this biological knowledge into genomic prediction schemes. When you look at the integrated functional annotation map of BovReg, you see the different layers which we addressed, from transcriptome down to metagebasis. And the idea was that we want to filter across all these different layers of information, so then in the end to identify variants which we think might have a particular emphasis, a particular impact on the regulation of the bovine genome and also the respective phenotypes, to be then channeled into biology-informed genomic prediction models. Within those layers of different functional annotation, EQTL is one of them, and this is also part of the BovReg project. For EQTL, we frequently face challenges regarding bioinformatics, because usually for a decent power you need quite a lot of data sets and also quite heavy data sets, which creates a huge computational burden for these pipelines. In addition, because you need many samples, it's usually a multi-partner approach where there are sometimes restrictions regarding direct access to raw data. And finally, when we want to channel these results into routine genomic predictions, you would like to have a high reproducibility of these pipelines to be able to rerun them repetitively in the same way. And to overcome these issues, workflow managers and containers would provide a good solution, because workflow managers are scalable and reproducible, and the containers, they provide the means to run all the necessary elements of the pipeline in an any computational environment. So Praveen Chitnedi, a postdoc in my lab, he developed EQTL Detect. That's a container and workflow manager based EQTL detection pipeline. And then 5 partners from BovReg contributed genotype and phenotype data. So genotype data, these are biallelic variants imputed to whole genome sequence level from 4 different populations, covering beef as well as dairy. And for the RNA-seq, we had raw data sets for 8 tissues, again, tissues which we think are relevant also for the target traits of BovReg, which I mentioned in a minute before. So taking Docker and Nextflow DSL tools, Praveen developed a complete workflow starting from the raw RNA-seq data sets until providing the final tables of output after filtering for statistical significance after permutation. This pipeline is also available, as mentioned already before, for some of the gene switch pipelines for download in the EuroFAANG data portal, so you can immediately go there, download and test how they work for your environment. EQTL Detect is comprised of several modules. You can see it's really a comprehensive, very complicated pipeline, but it's very easily substructured into 5 modules, which act as sub-workflows, which can then be distributed either across different servers or distributed across different partners. It also provides the opportunity to rerun the pipeline from different entry points when you want to modulate some of the parameters without the need to rerun the entire bioinformatic pipeline itself. We did develop the pipeline for gene level, for gene expression level, transcript level, and also splicing event level. And we took benefit of the new transcriptome annotation in BovReg, which Gabriel Costa Moreira will talk about in a few minutes. So we have about 44,000 genes, which we considered at gene level. And depending on the tissue, we had between 500 and 6,500 genes, which show at least one significant EQTL hit. As a proof of principle, we looked into the data in the literature, whether we could find some of the EQTLs which have been frequently reported also in our data sets, and it showed that, for example, for chromosome 6, for this very prominent GWAS hit for mastitis resistance at 88 megabases, we indeed found that the our lead EQTL for the GC gene is almost identical to, or very, very close to the one which was reported in the Lee et al. Study. So obviously we are on the right track there. We had the chance also to merge genome annotation, transcript classification, and these molecular phenotypes. And for example, we looked, are there EQTL hits which are consistent across several tissues, so finding them in several of those tissues we monitored. And for example, here is one which is significant in 40 different tissues, obviously being relevant for the expression of this MTBP gene. You can also see from the IGV view, can you see the mouse? That obviously this is a very nice enhancer region here, which matches this EQTL peak. Interestingly, another gene which is oriented in a head-to-head orientation in the same area is not affected at all by this genetic variant, showing that indeed there's a high specificity, obviously, of these EQTL signals. Another thing which we ask ourselves is, okay, we have the initial analysis showed one EQTL hit per, or filtered one EQTL hit per gene, but what is if there a phenotype is maybe modulated by more than one independent EQTL signal, or the other way around, are there SNPs which may be associated with more than one phenotype, so the expression of more than one gene. We looked at this in an area of 1 megabase space from the start of the respective feature gene transcript or splice site. And again, I show you the results at the gene level here for liver. On the left side, you can see that obviously there's a substantial number of genes which show more than one independent EQTL signal, sometimes even more than 4. Also, it's not an even distribution across the chromosomes. And on the right side, you can see that obviously there are some SNPs which affect the gene expression of more than one gene. Coming back to this annotation map, we also, besides the EQTL, we looked at the epigenetic response to maybe provide some biology-informed information for management. And for this, we looked at the intergenerational consequences of environmental stresses and took seasonal temperature stress. I, on purpose, wrote down seasonal temperature stress because the temperature we reached were 70 TIH. And I think people in Florida, for example, would just smile and not term this a heat challenge. So I term it different temperature stress. In this model, we had 3 different sires for which the semen was sampled either in winter or in summer. So in winter, you can see that that TIH was about 40. And in summer, you see it's northern Germany, it was just reaching 70 and not more. But at least there is substantial difference. And then we looked at what we call summer sperm or winter sperm. So remember, it's the same sire, just the semen collected in summer or in winter. We took the semen and produced F1 embryos from these, and then transferred the embryos, grow them to mature animals, collected semen samples again, and used this for creating the F2 blastocysts. And we looked at the phenotypes. We were a little bit surprised that we didn't see any effect of the F1s. So the semen, whether it was collected in winter or summer, didn't show differences in motility. However, when we looked at the sons born from winter or from summer sperm, there we could see significant differences in motility, showing higher motility for the winter sperm created animals, and this would consistently reproduce in 2 independent samplings. So obviously there is an effect on the phenotype. And also we looked at the embryos which were developed from these sperm samples, and for the F1 embryos which have been produced by the F0 sires, we see again a significant result, a different result regarding blastocyst rate at day 8. And this is statistically really significant for the F2 embryos created from the F1 sires. We see the same trend. However, at the moment it's not statistically significant, but we are following up on this. We then also looked at the embryos and looked whether there might be epigenetic effects from this mild temperature stress on the genome RBS methylation pattern in the embryos. And we found that obviously across all chromosomes consistently, except one, that's chromosome 28, we see consistently a hypermethylation of the embryos generated from winter sperm compared to summer sperm. So obviously there is, even with this mild temperature stress, an effect which you can see in the F1 generation. So this is a little bit about the different work packages. So in one work package 4 about the EQTL work, and work package 5 about the environmental sensitivity. And in the next presentations, you will hear something about the functional and structural genomics annotation from Gabriel, and about the biology-driven selection then from Renzo. With this, I'd like to also show our BovReg team at work, and really thank for an excellent collaboration, all the different partners who really worked hard to make this project happening, and particularly in red, mentioning those colleagues who contributed most to the EQTL and the environmental stressor research. And with that, I'd like to conclude and maybe forward the baton to, I think, Renzo for the next talk.